Hello, everyone, and welcome to our first Mind Matters webcast of 2022. We are going to learn if absence makes the heart grow fonder. I'm Miriam Good, CEO of Mind Science Foundation, and we fund neuroscience research and education. I'm so glad you're here tonight. Our guests are the 2020 BrainStorm Neuroscience Pitch Competition winner and postdoctoral neuroscientist, Dr. Merridee Lefner, and Dr. Matthew Wanat of the Wanat Lab at UTSA, University of Texas at San Antonio. Doctors Lefner and Wanat are going to share what they've learned as a result of their Mind Science funded project about if and why absence makes the heart grow fonder, which is really a look at the neurobiology of motivated behavior. So welcome, Merridee and Matt. And I'm going to start with you, Matt. And first of all, I wanted to give a shout out to UTSA for attaining what I always thought of as, what I saw advertised as tier 1 research status. And can you talk a little bit about what that means for UTSA and about your lab and why you do what you do in your lab?
Oh, that's well. First, thanks for having us. This is really great that you're putting this on. So yeah, everybody here is excited at UTSA. They've reached its Carnegie R1 level institution, which really just means a lot of research is going on here, and we're in with sort of the other big universities, and I think one of only 2 Hispanic serving institutions that have that designation, and so it's really exciting times here at UTSA. And
super.
Yeah.
Super. Can you talk a little bit about the work that you do in your lab and how Merridee came to your lab so we can understand, Mind Science has a focus on funding early career researchers like Merridee. And it's been interesting for me to learn, the path of a young neuroscientist. And we'll talk to Merridee as well. But from your perspective, Matt, how did she come to the lab, and what kind of work have you been doing together?
Well, so the lab in general, we're really interested in trying to figure out why we do the things that we do. And I would sort of argue a lot of our behaviors that we engage in can be grouped into one of 2 major categories. And the first of which is positive reinforcement, which in layman's terms is seeking out good things. And so say for example we want a piece of chocolate cake, we can't snap our fingers and it magically appears. Life is not fair. We're gonna have to get into a car, go to the grocery store, buy some cake mix, follow the instructions, and only then will we get our rewarding outcome. So that's positive reinforcement. But the other major group of actions that influences our behavior is negative reinforcement, and that's engaging in actions to avoid bad outcomes. And so say we're following the instructions of making the cake mix, and the oven timer goes off, and we stick our hand in to pull the cake out, and we're going to burn our hands. But what you can learn to do is if you put an oven mitt on, you're going to prevent that bad outcome. So that's negative reinforcement. So we're really interested in the neural systems that are involved with how we seek out good things and how we involve bad things or avoid bad things. And there's a number, while that seems sort of intuitively quite simple, there's a number of neurobiological processes that underlie these behaviors. So you need to involve learning. So learning what course of action gives rise to these outcomes. Motivation plays a role, but most importantly, decision making. And we've been interested in this idea of how does time potentially interact with our decisions, and how does that sort of change our behavior? And really sort of getting back to the other part of the question you were asking, this is sort of a crazy idea, had when, sort of starting up the lab is, does absence make the brain grow fonder? And we thought that, this neurotransmitter dopamine was going to be involved, and, Merridee can talk about how we were wrong about that. But, Merridee, joined the lab as an undergrad and, got started on this project. And it's really been fantastic to see that, Merridee has carried this through, and, really a Herculean effort of, having sort of a crazy idea. How do we come up with a behavioral task to test this idea and get some cool results, and then can't figure out what brain regions are involved, and then ultimately through a lot of blood, sweat, and tears, figure out what's actually what brain regions are playing a role. And so it's been fantastic that Merridee's been in the lab from an undergrad, as a research technician, graduate student, and now a postdoctoral fellow just for a little bit longer, as she's gonna, flap her wings and move on. But it's been a great having Merridee sort of take this from the first step of the project to its fruition to where we are now.
That's wonderful. I love to hear about this journey, the journey of a young neuroscientist. And Merridee, can you talk a little bit about, your journey at UTSA and with Matt, and why you're drawn to this particular line of research?
Sure. I will actually start a little bit further back, if that's all right with you, from when I was younger, because this is actually a long story in the progress. So when I was younger, I knew that I wanted to work kind of in the medical field. And then at some point in middle school, my grandma was diagnosed with Alzheimer's disease. And so that was really difficult for me to experience, watching her go through that. But it really made me curious about the brain and kind of changed the trajectory of what I wanted to study. And so then when I was in high school, kind of thinking about college, I ended up choosing UTSA because I had heard that there were, a bunch of great neuroscience courses offered at UTSA. And so I ended up doing a concentration in neurobiology and took like 5 or 6 neuroscience courses. And I learned from my professors, I didn't really know that this was an option to do research and have a career in academia and do research. And so that's when I decided that I wanted to seek out a research position and pursue graduate school. And so I ended up talking with one of my professors of my classes, and she pointed me in the direction of Matt's lab, thinking it would be a good fit. And so I was lucky. Matt let me volunteer as an undergrad in his lab, and then pushed me to also apply to graduate school. And, when I was choosing what graduate schools I wanted to end up at, I ended up choosing UTSA because with my experience of volunteering in Matt's lab, I just knew that there was so much more that I still had to learn from him. And I had started these projects that I really wanted to see through. And so, funnily enough, the first project that I had started in his lab was the one that we're talking about today, the BrainStorm project. And so throughout my PhD, I've grown to be really interested in the neuroscience of how we make decisions, specifically decisions about rewards, and how we can kind of change our decisions based on different factors, including, the time before a reward is delivered.
That's fascinating. Can you dive in a little bit into and tell us about the project and what you were seeking to find out and how you went about it and what you found out?
Yeah. So the main goal of the project was to test, what brain region is controlling a change in preference. And so we use the term absence makes the heart grow fonder because the meaning behind that is, if you go without something for a long time, you end up kind of having this longing for it. And that indicated to us that our preference for rewards are not set in nature, but we could actually change our preference. And that could be influenced by time. And so we studied this effect using rats, and we added a long wait time before a reward was delivered. And that ended up causing the rats to increase their preference for that reward option. And as Matt said, we're a dopamine lab primarily. And so we thought that the dopamine system would be driving this effect, and quickly found that we were wrong. And that's when we started to do a deep dive into the literature about what could potentially be causing this change in preference to occur. And I came across the retrosplenial cortex. I think at the time, Matt was asking me to look at a different brain region. And I said, please, can we also do some preliminary experiments in this brain region too? And he said, yes. And so that ended up being, we found some neat results. And, with the help of the BrainStorm funding, we actually, were able to fully flush out those results and get it published.
Wonderful.
And I just wanted to add, not only does this tell us, what part of the brain is controlling this change in preference, but kind of as a bigger picture, it's helping us understand, the cognitive processes that are driving our everyday decisions.
Can you talk about like the methods that you use? Now, I always preface all of my questions by saying I'm not a neuroscientist, so sometimes my language is more of a layperson. But I know, can you tell us, how you conducted the experiments and how what role the rats played? And I believe you had optogenetics involved. Can you talk about all of that?
Yeah. So with the preliminary experiments and the behavioral results that we found, what we did is we had rats in a preference test, and they could choose to eat chocolate and banana flavored food pellets at the same time. And so they got 10 minutes and they could eat whatever they wanted. And we just weighed out how much of each flavor they ended up eating. And that told us what their preferred flavor is. So if a rat ate more chocolate out of, all of the pellets available to them, then they preferred chocolate. And then rats went into training sessions where the initial less preferred reward. So let's say if the rat prefers chocolate, the less preferred reward flavor is banana. We deliver that one after a long 60-second delay. And then in separate sessions, the preferred chocolate flavor was delivered after a short 30-second delay. And we found that with training sessions, we then did another test and found that the rats actually increased how much of the less preferred or banana flavor they were eating. And so this indicated that this long delay before the less preferred reward increased preference for it. In the initial trying to find out the brain regions, we actually just did lesion studies, which is pretty basic old school science, where we just kind of inactivate all of the neurons in that brain region to really just find out if the brain region is required or not. And then some of our future proposed studies would be to use optogenetics, which uses a light to either activate or inactivate specific groups of neurons.
Great. So what does it mean, or what are the implications if you're able to use the elapsed time to increase a reward or increase desire for something that was initially less preferred? What does that tell us?
So that tells us that that time can definitely influence reward delivery influence reward value. And, it follows the absence makes the heart grow fonder idea of, having to wait for something or having some kind of cost, such as time, added to a reward can increase its value for it. And you can use an example of maybe you're waiting in line for something as well. And by the time you get to the end of that line, you have such a relief that you're finally at the end, and you've invested this time in the line. And so that could make the outcome potentially more rewarding to you.
Okay. Yeah, one of the things we sort of hope to maybe use this information going forward is in the context of substance use disorders, and they're often characterized by, individuals sort of, wanting or, a particular drug reward over, say, a food reward. And, one of the hopes is by identifying sort of these brain systems that might be involved with regulating your preference, can we devise strategies to specifically target these neural systems and be able to sort of change one's preference so that you can, reverse some of these maladaptive behaviors you might find. And so that's the larger mile-high view of hopefully where we could get. And I think one of the exciting things about Merridee's project is she's identified sort of a brain region in the retrosplenial cortex that hasn't garnered nearly as much attention in sort of the context of reward valuation. It's sort of a brain region that's been classically studied in the thought of, spatial navigation. So where are you in a room?
Oh.
And there is some increasing evidence that it plays a role in, timing related learning, and also some studies that, which were pretty much the reason why Merridee wanted to go down this road, saying that there are some, neurons in this retrosplenial cortex that respond to rewards. And so, putting that all together, Merridee was like, wait, there's a brain region that's involved with, timing and involved with reward valuation. Why don't we try and see if this brain region might be mediating this sort of interesting behavioral phenomenon that she characterized?
Oh, wow. And so I think I heard you say, Merridee, that you thought that dopamine would be involved and it wasn't. Am I saying that correctly?
Yes.
So is dopamine ever involved? And can you talk a little bit for people who don't know, can you give a brief, 60-second primer on dopamine and how it typically works in the brain and what its function is?
Yes. So dopamine neurons respond to rewards, both natural and drug rewards. And dopamine is highly involved in something called delayed discounting. So it's actually the idea of time negatively influencing a reward. And so that's why we thought that the dopamine system would, be involved.
Oh, okay. Wow. So can you talk a little bit more about, Matt, about a little bit more about the retrosplenial cortex? And you mentioned that it's involved in time, the perception of time.
Yeah. So, this is one of the challenging things, sort of when talking about this is, it's a big brain region, in essence, because it runs sort of like a mohawk, if you will, right down sort of the midline, sort of top of your brain there. And so, any one brain region isn't going to have just one specific role. And so, what we know about it is, from prior studies that have, looked at it in sort of trace conditioning, is this idea of you have a cue and you have to remember that cue happened, and then there's sort of an outcome. And so there's a gap in time that's important, that you need to be able to connect. Hey, there's some sort of cue, and then there's reward, and you want to link those two together. The retrosplenial cortex is involved in that, and so that's one of the functions it's been involved with, and then also in sort of being able to remember where you are in your surroundings. And so those are sort of a lot of the classical functions. And when I say classical functions, it's really just because that's where the science is and what people have studied it. And so we're really excited to go forward and studying the retrosplenial cortex in the context of changing reward preference, which is sort of a new spin that it fits in with other prior research, but I don't think people have really been looking at it in sort of the angle we're taking on it.
Right.
So.
Oh, that's great. And Merridee, can you talk? I brought up optogenetics, and you said that would be in a future iteration or the next step in the study. Can you define optogenetics for those in the audience who might not know specifically what that is, and then how, what a wonderful gift it was to research to be able to use optogenetics?
Sure. So kind of the idea of how optogenetics works is it's a virus that you can inject into certain brain regions, and it essentially inserts a channel into the membrane of a cell. And so you can use light to open that channel, and you can allow either positive or negative ions to flow through it, depending on the type of virus. And that would either turn on the cells or turn off the cells. And so we can use this as a way to, activate or inactivate neurons within specific regions just by shining a light on in the brain. And so, future studies, it would be really awesome to, start implementing optogenetics specifically within this project, because then we can start studying, which neurons in the retrosplenial cortex by turning on and off specific groupings, as well as looking at projections maybe between the retrosplenial cortex and other brain regions as well.
Okay.
And that's actually one of oh, if I could jump in there and promote a little more of what Merridee found. So she actually found in this study that the retrosplenial cortex isn't the only brain region that's responsible for mediating this, enhanced preference for an initially less, preferred reward. And she found that there's another brain region, the basolateral, the amygdala, that's also required. And so one of the cool things about optogenetics is you can be able to specifically target and turn on and turn off and manipulate the activity of, say, the cells from the retrosplenial cortex that go to the basolateral amygdala or vice versa. And so this is a way that you can really put together a circuit and say how brain cells in one part of the brain talk to cells in another part of the brain, and if they're responsible for mediating any type of behavior. And so it isn't just the retrosplenial cortex, it's also the basolateral, the amygdala as well. And so there's a lot more there.
Right.
And I will say, I think that the basolateral amygdala is the brain region that Matt was trying to get me to study when I, begged to also look at the retrosplenial cortex. So we were both right in a sense.
I love it. And so Merridee, what are next steps for you in your journey? If you're comfortable talking about what the future holds for you.
Yeah, so I am currently a postdoctoral fellow in Matt's lab still, just through March. And I'm currently looking for postdoctoral positions, across the country. I have some prospects in mind, but really I just want to end up in a lab where I can continue studying how reward-based decisions are influenced by things like time or, the size or flavor of rewards, et cetera. And, hopefully diving into other brain regions as well, using, techniques that are novel to me is what I would hope to learn.
Oh, that's wonderful. It's very exciting to have seen, you were kind enough to invite me to watch the Zoom of your dissertation, which was a real pleasure and honor. And it really gave me a sense of your whole, like the arc of your journey. So I was really thankful for that. And congratulations.
Yeah, well, thank you for coming.
Yeah. And so I'm going to, I think I'm going to throw it to Q&A. And I'm going to start by going back to that question about what a possible intervention might look like, like the ultimate, in years hence, would an intervention for addiction, say, would that be based, like, would that be something where you would physically do something in the brain, or would it be like a medication you would take? Or I know we're not going to do optogenetics on people, but what form might that intervention take?
Well, I think one of the hopes about this project was that maybe we could just have a behavioral intervention, and that's if we can devise sort of, behavioral strategies where we can, alter one's preference for, what is a sort of a rewarding outcome that potentially leads to, maladaptive, behaviors, and switch that to, something that is, not, as, leading to problematic behaviors. That's, I think, the ultimate goal. And, devising behavioral strategies is great. But also, yeah, I mean, we don't, by identifying the neural systems, sometimes there are unique characteristics that you can find in some neurons and part of the brain that maybe some pharmacological interventions will specifically be able to tweak the activity of those neurons so that you can get the desired behavioral outcome. And so it's sort of hopefully a 1-2 punch that, we can identify behaviors, the neural circuits as well, and maybe also there can be, pharmacological strategies that we can identify as we go on this journey of figuring out how the brain works.
Oh, great. So I'm going to, like I said, I'm going to throw it open to questions. We have a few minutes here left. If you want to go to the Q&A box or even chat if you'd like. So I think one thing that, and is really interesting for me is going back, and again, decision making in a larger context. What is your lab, apart from this study, what else is your lab working on right now? What are some of the other things to do with decision making?
Well, we actually have sort of a number of different projects in the lab, and I guess maybe to highlight maybe one of Merridee's ones that we're working on now is looking at sex differences. And I think this is something that has clearly been understudied. It's great that there's now some renewed intention, but we're interested in sex differences and behavioral responding to cues that are predictive of reward, and looking at the neural signals and how they're sort of different between males and females. And there are interesting, differences. Males and female brain, they are not the same. And in some ways, you might actually sort of think of it as a spectrum, if you will, and not necessarily a binary, split between there's a male brain and a female brain. But you can see sort of it is sort of a spectrum across as far as the heterogeneity they'll see in both behavior and also the neural signals that we're looking at.
Mm-hmm. And you talked earlier, and I made a note about so you were talking about learning how to use an oven mitt instead of taking the pie out with your bare hands. And I assume, as anybody who's ever parented a toddler or an infant, we know that starts early, that learning starts early. But how early, and are there things that parents can do to increase the effectiveness of that time or that experience with young children?
Well, in general, it's sort of known that positive reinforcement is better than negative reinforcement. So, sort of guiding behavior and saying you did a good job after something is going to be much, better than saying, don't do that, don't ever do that again. And so that's sort of a better way, even though sometimes it can be challenging to sort of catch yourself and say, no, let's be positive as much as possible. But so from that's from an early age, and it is actually interesting that we can use rodent models to be able to study these things, and the interesting processes of what neural systems can regulate how we learn about seeking out good things and avoiding the bad outcomes as well.
Great. So we've got a few questions here. From Beth, she says, great work. You mentioned the retrosplenial cortex had previous implications in spatial memory or reward, whereas your study focused on timing. Is it possible it is also involved in food-specific reward? And could future studies compare or isolate the influence of timing versus food versus spatial reward?
You want to take that, Merridee? It sounds like one of
sure. Yeah, I think that was definitely something that we've been thinking about as well, as far as moving forward, is moving around the locations of the rewards before and after the training sessions to see if when we inactivate the retrosplenial cortex, does it affect our results depending on if we're changing the location of the reward or not, or stuff like that. But as far as the flavor of food reward, we actually had a mixture of rats as far as which flavor they were actually preferring. And there was no effect of the lesion studies, at least, on the prevention of the change in preference, depending on which flavor food reward was preferred or not. So, yeah.
It's also a funny story with that. So, us being scientists in the lab, we wanted to be academic about which flavor food that we liked. And so the banana flavored food pellets sort of taste like stale banana runts. And the chocolate flavored food pellets sort of taste like, stale Cocoa Puffs. We went around the lab, and about, 50% of the people like the banana flavored food pellets, and 50% like the the chocolate, and even did an unofficial Twitter poll and asked what people wanted. And in general, the scientific community was split. 50% banana. Actually, I think it was 40% banana, 40% chocolate, and 20% are saying, "Why are you eating rat food pellets?" But that's.
I love it. I choose chocolate. I may be mispronouncing this name. Rainier asks, is there a correlation between delayed reward and patience? Not sure how to test patience in rats, though.
Do you want to take that again, Merridee?
Okay, yeah, again, I don't really know, I guess, how you would test patience, except if the rats had to actually choose to wait for the reward. And there has been studies showing that rats will choose to wait for rewards. So the task I'm talking about in particular, the rats had different rooms that they could go to and choose to wait varying amounts of time and receive various amounts of or types of rewards. And so rats will actually voluntarily, wait longer times for rewards that they actually want more. And so I guess that would be a correlation between, patience, or, a potential correlation between patience and delay, but it's not necessarily, one-to-one, where a higher delay equals, more willing to be patient, I guess.
But that does bring up an interesting point where you had also found that you looked at the anticipatory responding. So I guess it's not necessarily patience, but in these training sessions where the animals didn't have to do anything, they would just get a food pellet, and we could track, how the animals were anticipating it by looking at the sort of the number of head entries that the animals were making beforehand. And really, there was no difference between the anticipation for the reward that was delivered after a short period of time with a long period of time. And, when you lesion those brain regions, it didn't have any impact on that. So it seems that at least anticipation, which isn't the same as patience, but was not related to the ultimate change in reward preference.
Okay, and Manuel would like to know, do you believe this research has relevance for changing preference between something that is liked and something that is disliked, or does that use a different mechanism than the most liked, less liked relationship that you used?
Yeah, so I think that is kind of similar to what Matt was mentioning earlier, where the dream goal of this project would be to see if we could change preference for, something like a drug reward to a more natural reward, or even, maybe changing preference. I think you were saying this earlier, Miriam, that changing preference for ice cream to like a carrot or something. And so I do think that this could apply to, changing preference for a less, a nice reward to a less nice reward. I don't know, how much you have to like either reward in order for us to see this effect, though.
Yeah.
But I think it's a good point. I personally don't know if you could be able to make something that you don't like and then do this behavioral manipulation, and then you're going to like it. You have to have some sort of intrinsic want to sample it, because if the animals aren't sampling it, then they're not going to be able to associate the time with the actual rewarding outcome. But that would be sort of fantastic in a way to be able to do that, of, going back to, Miriam, as you're saying about the kids, get them to eat their vegetables. I mean, I know one of my kids won't even touch vegetables, and, that would be fantastic if we could, finally replace the chicken nuggets with some broccoli on occasion.
We can dream. Well, thank you both so very much. It's always a pleasure to be with you. And just a reminder to our audience that our next webcast is on March 29th, all the way live from Paris, France, an interview with another BrainStorm finalist, Lorenzo Ciccioni, on his project to increase graphicacy, which is literacy for being able to read the myriad of pieces of information that come to us every day in the form of graphs and charts, and how critical that is over the past several years in understanding this flood of information that drenches us each day. And then on May 19th, we'll have our first in-person speaker event since 2019, featuring last year's BrainStorm winner, Ben Ryan, who will talk to us about his work exploring how early experiences shape the social development of our brains. Dr. Ryan is also a science influencer with flourishing audiences on TikTok and Instagram. And you can keep track of our events by visiting our website on a regular basis. And so, Merridee and Matt, thank you again. It's been just a joy to be with you. Thank you for helping us understand this region of the brain and how these reward preferences work. Thank you again. Good night, everyone.
Thanks for having us.